Hi, I'm Alan Hoyle from the University of North Carolina Lineberger Comprehensive Cancer Center, and I'm here to talk about jUNCtion, which is a tool that we wrote here. So we have an eternal struggle here. Investigators have their data isn't useful without analysis and processing, so how do we help our investigators get their data processed through workflows? So here's a little bit of an outline. I'm going to touch on this as we go through. So first of all, who we are and who we support. Our investigators at the Cancer Center, they vary from distinguished professors. We support a few dozen labs. We go down to grad students. Some of them have extensive programming experience and their own bioinformatics teams, but some of them have also never touched a command line. The projects we have cover a wide range as well. So we have large multi-center international collaborations with thousands of subjects like TCGA, and we also have small projects that just come from a single trainee. The data we have comes from two sequencing centers, the campus-wide HTSF and our Cancer Center Translational Genomics Laboratory, and also external databases or external repository data that we download. And the LIMS we have has registered more than 7,000 more than 7,000 flow cells and 180,000 FASTQs. In our group, we have about 20 people. We have half a dozen PhD analysts and other workflow and production people who run workflows. We have our LIMS team and an admin team, and about half of us, like I said, run those computational workflows. The LCCC has a compute cluster. It's medium-sized. We run Slurm using Singularity as the background, and there are also other campus resources available. So where we came from and how we got to where we are. We wrote our own workflow engine a while back called RAP, Workflows for Rapid Analysis and Pipeline Processing, and it was a bunch of spaghetti Perl scripts that took bash-like scripts, converted them into a bunch of separate jobs, and they ran sequentially on our cluster. And then the next one would be kicked off by a cron job as they finished. So it worked. We ran tens of thousands of samples through it, but the workflows were linear and fragile. The workflows only ran per sample, so any kind of cohort-wide summaries had to be run manually, and our group had to run everything for everyone, so we had less time to tackle new problems and writing new workflows. So running workflows was a full-time job for 2 or 3 of us. We dabbled in CWL to get away from RAP, and we learned a lot, and part of it was that we didn't like CWL very much, but we did like the features of applications like Orvados and Curaverse and the things they offered us. So with our workflows, we learned that we can advance a lot of our science by running generic curated workflows to a whole bunch of data sets. So to keep this going forward, we looked for a new workflow engine to replace RAP and CWL, looked at Toil and SnakeMake, and one of our developers found Nextflow, and that's what we've done ever since. The current Nextflow workflows we've developed are an RNA-seq quantification, DNA-seq somatic and tumor-only and tumor-normal pairs, some single-cell RNA, and some lesser workflows that we've done. So we have a selection of workflows here. So we don't want to run them all ourselves, so how do we do that? So what we did was we created jUNCtion, and that's a tool that lets all of our investigators run the workflows on their own data without having much training involved. It's a web application. It's Java, JavaScript, HTML, Python, and Bash. It's heavily integrated with our local infrastructure, so our campus SSO, our LIMS, and our LCC compute cluster. So data comes in from the FASTQs from the various sources. It gets loaded into our LIMS. jUNCtion, when the investigator comes in and creates something, they query the LIMS, create a data set, they run that on the cluster, which then saves to the file system and also saves information back into the jUNCtion database. So it's a modern web interface. Our standardized workflows can be run, like I keep saying, with little training, and it has tight integration with all of our local stuff. We can also share results with labs and other users. Our permissions are much more flexible than what we had with our regular file system, and monitoring and reporting are centralized, and it helps us find and help with problematic processes when something dies. On the other hand, we have local development time for any features. The Unix permissions still come up for advanced users, and updates can be complex because we have many tech stack dependencies. So when someone gets to jUNCtion, here's what they see after they've logged in and selected their lab. Down on the bottom half of the screen, we have sort of the what's going on right now. We have news and documentation, we have the currently running workflows, and we have the recent results of workflows that have been run. But a lot of times when they come in, they want to get to work. So the first thing they have is they have the option to manage their studies, they can execute their workflows, and they can view the results from previous workflow runs. So when they are here to create a study, they can go and do a query from our LIMS system to get the sample metadata, or they can upload a TSV manifest. And then when they know that that populates the study, and if they know there are specific samples that are bad, they can remove them by selecting things over here. And we can also do merging existing studies. So our workflows run in two modes. We have a per sample mode and a summary mode, which applies to the entire cohort. So if a if you've already run one flow cell's worth of data, and a second flow cell becomes available, you can just take that first flow cell, combine it to the second flow cell, and then run that as a single later combined data set. So this helps to maintain provenance to avoid redundant computation and storage. Now, once you've created it, then they can go and execute their workflow. So they come in, they click on that button, they select the data set that they want to run their workflow on, and then they select the workflow that they want to run, and that includes the reference genome and annotations or capture versions, and the Git tag so that we can reproduce this outside of jUNCtion if necessary. They select a few other things, like whether they want email notifications or publishing BAMs, and then they click the Start Workflow button, and then on that front page, something like this appears. And that's really important because we get the question a lot, where is my data? And so we implement part of the jUNCtion Web API to get data from the Nextflow process into jUNCtion, and we also expose the Nextflow logs and trace to the app and user. And when the workflow ends, if they've selected that mail thing, they get an email telling them that their workflow has successfully succeeded or failed. Then, since we've created all of our own workflows, we have a standard output format and data structure, and we display the results through the web with a custom JavaScript and HTML templates based on our standards. So we get the per study information, and the per sample information is down below. Thanks to other projects like IGV, we've integrated things to allow our investigators to look at their data in more detail without having to download many gigabytes of files to their PCs. Advanced users, though, can use our cluster's VMs and access the data directly through and do things with more traditional methods, like they can bring up R and IGV and the native IGV on the cluster. We also have other features that allow us to centralize our reporting. So we scrape our logs, we generate monthly and annual reports so we can find out our file system and compute impact, and the cluster usage for the workflows and for the labs. And this is also useful for getting information. We get the questions for, what do we need, what happened for a grant or a paper. Over the past 2 years, we've run over 1,200 workflows, 48,000 samples. We have 60 labs and teams. Some of our labs are actually multi-lab collaborations, and 130 users. And just last year, we ran a TCGA scale thing locally on our compute cluster. So we have some future improvements in mind. We want to modernize our back end, and we want to be able to containerize the application for easier development. And this is the short and medium term things. In the longer term, we want to allow Cloud Executor with provisioning, increase the customization on workflow runs, so more user-provided parameters and references, and maybe NF core and community workflows. We want to be able to chain the workflows together, add remote sharing via Globus, and release our source code to the community. But in summary, though, jUNCtion allows our investigators to log into a web application, create a study with their data, run it, and review the results, all with relatively little training. So I'm welcome to have open for questions. The current jUNCtion developers are Con Liu and Ben Wingo, and former developers John Ford and Ryan Latta. Chuck Peru and former director Joel Parker came up with the idea for jUNCtion, and our current directors are Sai Balu and I accidentally left off my other current director, David Corcoran. And we were funded through the National Institutes of Health and the North Carolina UCRF. So I'm happy to take any questions.
Thanks for the talk, Alan. We have a few questions here. So Francisca is asking, how do you handle data safety for sensitive human data?
So since all of our data is saved on our local storage system, we just handle it the way we would handle any other data on our local thing. The jUNCtion is limited. It's behind our firewall. The permissions are all handled via the database, and it's limited to only certain people can access certain things through there.
So how difficult, so Phil is asking, how difficult would it be for others to run this software? How difficult would it be to make it agnostic to infrastructure?
So the executor part of it is a separate Python script that we intend to, that can be pulled out for running other things. When jUNCtion was originally developed, it was developed with CWL in mind. We could, in theory, add wrap or other things. And the toughest thing would be integration with a LIMS system. It already if you have a local LIMS system, you can already upload a sample sheet essentially to generate a data set, and that would be relatively easy. But there would be significant development in integrating with your local other resources.
So one last question from Evan. So what do you use for LIMS?
Our LIMS is a custom developed thing by our local group. It's more than a decade old, and they have a whole team doing development on it, and that's a Java app, and that is currently being ported over to a JavaScript app native.
Okay. So I just actually saw that we have time for one more question. So Yabo is asking, I think Mendilab can use jUNCtion. Would it be open source?
It will be eventually. We need to update the tech stack to make it more shareable. It's currently very old tech stack with like Java servlets natively.
Thank you very much.